Guys, this is a very exciting lecture. And why, you may ask? Because this lecture is about protein synthesis. Now you're probably like, okay, nice. Well, yes, it is. It's called protein synthesis. Doesn't sound real exciting, but guys, this is the secret of life. All your questions about biotechnology and cloning and all this kind of stuff that's going on, it all comes down to protein synthesis. Now, protein synthesis is what we call the central dogma in biology. Now, what does that mean? That means, again, the secret of life. This is where we are going to learn how DNA codes for RNA, which codes for proteins, which codes for traits. Now, what does that mean? That means your DNA molecule, which you've already learned all about, that's inside each one of the cells, 75 trillion cells in your body, it contains combinations of nucleic acids called nucleotides or nitrogenous bases, A's, T's, C's, G's. You've already learned that. Well, those things are going to code for making a molecule called RNA. And RNA codes for making amino acids, which code for making proteins. So you put the amino acids together, you get proteins, and it's these proteins that give us the traits. And you are going to learn exactly how that happens. Now, let's take an example. Many of you are familiar with mussels. Some of them taste really good, but they're very interesting organisms. And one of the things that makes them so interesting is that they stick to rocks and docks and whatever else they feel like sticking to. Okay, so what? Well, how do they do it? How do they stick to these rocks in water, in waves, and never fall off? How? Well, they produce a protein called bisus, and that bisus is like the world's best super glue, only better. In fact, scientists are so intrigued by bisus that we want to learn how to make it, because this could have all sorts of applications. Like, say you sever a nerve, maybe that bisus could be used to repair that nerve because it's waterproof. We've never been able to make a super glue that is waterproof the way these mussels have. Well, okay, so we're going to make this bisus glue. How are we going to do it? Well, we're going to take the gene from the mussels, clone it, and actually use it to manufacture the muscles bisus protein in the laboratory in large quantities so we can actually use it. How do we do this? Well, with the knowledge of protein synthesis. So let's learn how all this is done. Let's think back to proteins. You guys have already learned about proteins. You've learned that they're the most diverse molecules out there and that they play all sorts of different roles in our body. So you've learned that they act as enzymes, and enzymes themselves are very diverse. They can digest things, they can speed up reactions, all sorts of stuff like that. So speeding up reactions, lowering the activation energy. They have structural functions like hair, like the keratin of feathers, like the keratin of hair, etc., etc. And they can function for transport. So recall that hemoglobin, for example, will transport oxygen to our blood cells. So all sorts of things like that. But recall that no matter how diverse the proteins are, I mean, there are hundreds of thousands of different proteins all functioning simultaneously in your body right this instant. Yet, despite the diversity of proteins, they are all variations on a theme because they're made up of variations of just 20 different amino acids that exist on this earth. So pretty amazing. And you might recall, here's an example of what a protein might look like. This is hemoglobin, because you can see the heme group right in the middle there. And each one of these little bubbles might represent a section of an amino acid. So we can say we put all life on Earth on one slide because here are those 20 amino acids. And again, it's just adding these guys together that make proteins, and proteins do all those amazing functions. And looking a little closer here, notice how similar these amino acids are to each other. They all have this COO- group right here. It's a carboxyl group right there. They have NH3. They have a backbone carbon right there. So no matter which of these 20 amino acids you look at, part of them are all the same. And so it's just this R group that's attached onto them that is different. And so here's phenylalanine. You might have heard of that if you've ever picked up a diet yogurt, for example, and it'll have a warning to PKU patients, phenylketonuria, because they lack the enzyme to break down this phenylalanine here. Notice how some of these amino acids are very similar to each other. Like the only difference between valine and leucine is this one CH2 group right there. Notice, let's see, let me find asparagine. Here it is. Asparagine, it happens to be the amino acid that makes asparagus cause that smell the day after you've eaten it, if you know what I mean. So, and notice that all of these amino acids have a 3-letter abbreviation. So glutamic acid is GLU, for example. Histidine is HIS, etc. So they also have a single-letter abbreviation that some people use, but for today we're going to stick to the 3-letter designation. So again, just combinations of these 20 amino acids you see on the screen make all the proteins in the world. And those proteins make all the traits in the world. So just whether you're a bacterium or a human or an elephant or a tree or a worm, etc., we're all using these exact same 20 amino acids in different combinations to make the proteins that make us. Amazing. So how does this happen? Well, it happens in 2 main steps that make up what's called protein synthesis. So if you look at this cartoon right here, my goal is by the end of this lecture, you are going to understand each part of this cartoon and can replicate it in your own terms. So what exactly is going on? So these 2 main steps are called transcription and translation. So in transcription, we're going to take a section of DNA that codes for a protein. We're going to transcribe it, and that means rewrite it, into a molecule called RNA, specifically messenger RNA. And then you can imagine that that message is in a different language. So it needs to be translated during translation into an amino acid sequence that will create that protein. And that protein will give us our trait. So again, for 2 steps of protein synthesis are transcription, where we are writing the message, and this is going to happen inside the nucleus of the cell. So of course the nucleus is where you find your DNA, and this is where transcription is going to occur. And this is where DNA is going to be used as a template to create, to synthesize messenger RNA. So DNA, you already know, stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. And we'll go into some more detail about the structure of RNA later. Okay, and how is this going to happen? So DNA is going to serve as a template. Free nucleotides, remember they're floating around the nucleus all the time, they're going to come in and fill in to create the other strand after we've split the DNA. And that new strand is going to be messenger RNA. So you can kind of see that here. Here's your DNA. We're going to get another enzyme attaching at the start of a gene. It's going to be RNA polymerase. That, remember the weak hydrogen bonds, it's going to unzip the DNA into 2 sides. And free nucleotides floating around are going to base pair with one side of the DNA, and this will become the mRNA, the messenger RNA transcript. And that is carrying the coded message from the DNA. In other words, it has taken the gene that we need to create a certain protein, and it has written the directions on how to make it. But those directions are in a language that we need to still translate. And so that leads us to the second step called translation. And translation is going to happen in the cytoplasm of the cell. So how is this going to happen? Well, this is the more complicated part. So imagine right here is the nuclear envelope. Remember that there are pores in the nuclear envelope, and that's going to allow, after transcription, this messenger RNA is going to break loose here, exit through a nuclear pore into the cytoplasm. Okay, this is our messenger RNA, and this is where the fun starts. This is where we're going to start to incorporate a ribosome and another kind of RNA called tRNA, which will carry amino acids to create a protein. That's not going to make any sense to you right now, but I'm hoping by the end of this lecture it will. But in general, in a nutshell, translation, we're taking the coded message that started with the DNA, is now put into a language carried by messenger RNA, mRNA, and we're going to take it into the cytoplasm where it will be translated into an amino acid sequence, which eventually will create the protein that we need. Okay, so when you stick these amino acids together with peptide bonds, they form a polypeptide chain. And when you get to the end, you've created all the amino acids you need, then it will break off, fold up, and become your protein. Okay, so it'll fold into its protein 3D conformation. And if you remember, that really determines the function of the protein, is its 3D configuration. And recall that if you expose a protein to acid or to high temperatures, that will break those bonds between the amino acids and the thing will fall apart and not work. Could be a bad thing. Okay, so the basis of this is an absolutely magical code called the genetic code. And the genius who worked this out, you know, who knows. But this right here is what you call the genetic code. It's the magic code for life. Whether you're a tree or a worm or a human, we all follow the same code for creating proteins. In total, there are 64 codes within this genetic code, and these are called the 64 codons of life. So if you look here at this genetic code, you'll notice that there are 64 combinations, and these combinations are made up of 3 nucleotide bases. So, for example, the amino acid glutamate, its code is GAA or GAG. And here's how you read that code, and I'll go over this again in a minute, but you have the first base in that triplet, so this is the first base. The second base is right here, and the third base. So let's find G, okay, A, and the third one there is A. So you pull down and there's the A. So this is glutamate right there. But notice that GAG will also code for that. G, and then A, and then G, glutamate. Okay, so that's how you read the genetic code. So what's going to happen is you get every 3 bases in a DNA gene, in a code, will code for 3 bases on the mRNA. And mRNA, it's single-stranded, unlike DNA, which is a double-stranded double helix, but it's all going to be exactly the same except for 1 base. So DNA uses GTAC, RNA uses GUAC. So instead of thymine, RNA uses uracil, which is a U. Other than that, very similar molecules there. Now, all proteins are going to be made by starting off with the mRNA codon, that is a triplet of bases, call it codon, of AUG, OG. And we can say this is kind of the pirate's code, OG. So if you look on the code here, let's find A, U, that brings us down to this box, G. That codes for MET, which is methionine. So methionine is the amino acid that is called the start codon. So anytime we read AUG, that means that we're going to get a methionine as the amino acid that's there. Okay, so let's continue on here. Okay, so methionine is always the first thing in the mRNA transcript that will start a gene. And as soon as that polypeptide chain is made, and again, we're going to go over how this happens, when you're done, then you need a stop codon, which is 3 bases on the mRNA molecule. That means stop making more amino acids, and that will trigger some signals for the amino acid sequence to fold up. And there are 3 stop signals in the genetic code. UAA, so let's go UA, brings us to this box, and A, see, it means stop. UAG, so UAG, stop. And UGA, so UGA, stop. So there are 3 stop codons. And when you get to a stop codon, you stop. So think what would happen if you had a mutation in your DNA or in the RNA that coded for a stop codon instead of what you were supposed to get. So for example, let's say we were supposed to get glutamate made because we were supposed to get the mRNA codon GAA, but instead the G mutated to a U. We'd have a problem, right? Because then we'd get a stop codon instead of glutamate, and if that was early on in the sequence, stops everything, you don't get the protein you need, could be very bad. So UAA, UAG, and UGA are the stop codons. They don't code for an amino acid. They mean stop the production line. Okay, so that gives the instructions to stop adding amino acids to the growing polypeptide chain. So again, here's the genetic code again, just a bigger picture to make sure we're all on the same page. So just summarize, again, these are your 64 possible combinations to create these different amino acids. And these are just the 3-letter abbreviations for an amino acid: phenylalanine, leucine, serine. I don't expect you to remember those, but these just represent the different amino acids. And the genetic code is very redundant. So remember how we were talking about mutations in the previous lecture, and we said a lot of times you'll get silent mutations or neutral mutations. That means that, yes, you got a point mutation in one of those three bases, in the DNA or in the RNA, but it's still coded for the same amino acid. So it didn't affect the organism because you still got the same protein. So you can take an example here, leucine. There are 4 different ways just here to make leucine, and you can also make it a couple ways up there. So if you had CUU, you'd get leucine. But let's say you got a mutation, and instead of a U on the end, you got a CUC. No problem, you still got leucine. But now if the second base mutated, say instead of a U, you got a C, then you got a problem because then you get proline instead. So it just depends which base mutated in those 3 bases that codon.
The cell transcribes the genetic designs for each protein from strands of DNA to special molecules of RNA. An enzyme unwinds a segment of DNA, making room for its complementary RNA nucleotides. Both DNA and RNA nucleotides contain a sugar, phosphate, and base. But in RNA, the sugar is ribose, and one of the 4 bases is replaced by uracil. The exposed bases of the DNA strand serve as a model for the construction of messenger, or mRNA. On other strands, the cell produces transfer, or tRNA, and ribosomal, or rRNA. In their final form, these molecules play a vital role in protein synthesis.
This is the mRNA transcript now is about to leave the nucleus and will be ready for translation. Okay, so you just saw a little video on transcription. So let's just summarize again. I got some videos here from Pearson, and this will show, here's the beginning where the DNA is here, the RNA polymerase attaches to it, and we'll start unwinding it at one section. So you see the section here, and one side of the DNA will serve as a template for the mRNA transcript, which is occurring. So again, it begins with the RNA polymerase, which will attach at a certain point that dictates that there's a gene starting there, and that's called a promoter region. All it is a certain set of A's, T, C's, G's that enzyme recognizes as the start of a gene. So that's the place where RNA synthesis starts, and that gene is going to code for a particular protein. And then we get this growing RNA strand by just adding one nucleotide at a time to the DNA template, just pulling from free nucleotides floating around in the nucleus. Okay, so just again, recap, just remember that RNA uses uracil instead of thymine. So here's a picture of an RNA molecule, computer generated, of course. And it uses, instead of deoxyribose, it uses ribose. So other than that, it's single-stranded, uses uracil instead of thymine, and uses ribose instead of deoxyribose. Other than that, very, very similar, follows the same base pairing rule with the exception of using U instead of T. And there are actually 3 types of RNA. So we have messenger RNA, which is what we've just been talking about, that carries the building instructions for proteins. But there's another kind of RNA called ribosomal RNA. These are actually manufactured in the nucleolus of the nucleus. And ribosomal RNA is the principal component of ribosomes, which we'll be talking about in a minute because they're involved with protein synthesis. And finally, another RNA that will be involved with protein synthesis is called tRNA. This is also called transfer RNA. This is going to be what does the reading of the codons of the mRNA, and you'll see what we mean in just a minute. Just another little point here, which is kind of cool. Some differences between RNA and DNA in terms of differences between uracil and thymine. They think RNA actually preceded DNA over evolutionary time because it's simpler. And if you look at the compound uracil versus thymine, you can see they're very similar. Remember that there's a carbon at each corner here, so it's spelled out there, but these are the same. This has a methyl group on it that this doesn't have. But other than that, they're virtually identical compounds. So again, just like we were talking about in the previous lecture, what's the chance that during chemical evolution, we got a slightly different compound and the rest is history? Pretty cool to think about. Okay, so again, finishing touches with transcription. You get a cap at the beginning of the transcript, and that is just a modified guanine. So here's your cap here. And at the end, you get a modified, what's called a poly A tail. These are a bunch of adenines stuck together. So you'll get a cap, you'll get a tail, but one other thing actually has to happen before this mRNA transcript is ready to be translated in the cytoplasm. And that is we got to get rid of the evolutionary junk. So the evolutionary junk are called introns. The part that actually codes for the protein is called the exons. So not exon Valdez or anything like that. Exons, that's the coding region of the gene. The introns are all the other bases that have accumulated over evolutionary time that don't actually code for a protein. Well, we don't want a bunch of waste coming into the cytoplasm, so right before the mRNA transcript leaves the nucleus, that evolutionary junk is going to get spliced out. And that's what's kind of being shown if you guys can see this diagram right here. Imagine here is a coding sequence, so that's one exon. Here's another coding sequence, that's another exon. This was an intron, so it's actually going to pinch and get taken out. So that's going to be taken out, left in the nucleus, and broken down, and those bases can be recycled. So there'll be some of the free nucleotides floating around. Here's another diagram of this. So that's that non-coding region, that intron. And here's the exons, the coding regions. So again, this is your evolutionary junk. So introns, base sequences that get removed before the transcript leaves the nucleus. They stay in the nucleus and get recycled. And this is only in eukaryotes, by the way. Bacteria don't have this phenomenon. But this is our evolutionary junk. This will be important actually in a later lecture when we start getting into molecular evolution. That evolutionary junk is going to be important, and it's going to be important for an activity that we're going to be doing. So, but the exons get stuck back together. When you take the introns out, the exons will splice together, and that becomes your coding sequence that will eventually make our amino acid chain and our proteins. Now, before we get into translation, let's do a quick review of ribosomes, because it's these guys that actually will do the work. These guys are kind of like the factory upon which the assembly line will function. So here's a picture of a ribosome. Here's a picture of a ribosome. And so what you see here is this is your mRNA transcript that's going to be leaving the nucleus, going into the cytoplasm, and causing a small subunit of the ribosome and a large subunit to come together to make the whole ribosome. Kind of looks like a mushroom. And this mRNA transcript is going to go along through the ribosome until we get to the start codon, AUG. That AUG is going to attract a tRNA molecule, which you see here. And because AUG codes for methionine, as we found when we looked at the genetic code, so you always have to look back to the genetic code to find out what you're supposed to get, you'll see that that means that the anticodon to the codon, means the opposite, is going to be UAC. Because if you have an A here, then tRNA will have a U. If you have U here, tRNA will have an A, and if you have G here, tRNA will have a C. So this is the anticodon, this is the codon, and methionine is the amino acid that will be attached to this tRNA. And this tRNA settled into this nice little P site right here. There's open sites here where more tRNA molecules can come in and read the next codon. Okay, so 3 bases of the mRNA are read at a time, because that's what a tRNA reads. It reads 3 mRNA bases, a triplicate or a triplet nucleotide or a codon, reads 3 at a time. Okay, so basically translation is going to happen in 3 steps. The first is initiation, the second is elongation, and the third is termination. And so this is a model of a tRNA molecule, and it would carry its anticodon and its corresponding amino acid. This is a picture of the nuclear membrane with a nuclear pore here, and the snaky little mRNA transcript with its cap and its tail leaving the pore into the cytoplasm. And then it's going to meet up, and a ribosome is going to form around it, where every 3 bases are going to be read by the 3 anticodon bases of a tRNA molecule carrying its amino acid. So let's watch a video and see what's going to happen here. But basically, initiation is what gets started, elongation is the amino acid chain forming, and termination, you get to a stop codon, enzymes come and cleave the thing off, and it folds up into its three-dimensional protein shape. So let's watch a video.
Translation begins with the binding of a messenger RNA to a small subunit of a ribosome, the cell's workbench for protein assembly. When properly positioned, the messenger RNA triggers the approach of a tRNA, which carries the first amino acid. The tRNA attaches only if its 3 nucleotides exactly match the first 3 coding nucleotides of the mRNA strand. A large ribosomal subunit now joins the group to form a functional ribosome, with 2 binding sites built from ribosomal proteins and rRNA. Soon, another tRNA arrives, which matches the next 3 nucleotides on the strand. With the help of the ribosome and some cellular energy, the neighboring amino acids bond together. The first amino acid then separates from its RNA taxi, leaving the ribosome in search of another, identical amino acid. Now the ribosome moves along the mRNA strand. This exposes the next set of nucleotides, which match those of another tRNA. With each amino acid, the protein continues to elongate. As it grows, it folds into the 3-dimensional shape so crucial to its function. When the process is complete, the ribosomal fragments separate, free to join again later. With a legion of these molecules in operation, a single cell can produce hundreds of proteins every second.
Isn't 3D animation cool? Okay, so we're going to recap what you just saw in the video so you can learn exactly what's going on here. So let's start with initiation. So step 1 of translation, and remember we're translating the message that's coded in the mRNA molecule that left the nucleus, which ultimately was coded by the gene in the DNA. So DNA codes for RNA, codes for amino acid sequences, which create a protein, which codes for a trait. That's the central dogma of biology. Okay, so first thing you saw, mRNA came out into the cytoplasm, entered the small unit of a ribosome. Okay, so initiation of the small, it starts with the mRNA transcript binding to the small unit of a ribosome. And once you have that AUG, that start codon, R, that's going to attract the tRNA that carries the start codon amino acid, methionine, MET. So it's going to come with its anticodon, bind to the AUG of the mRNA on the small unit of the ribosome, and then the large unit is going to follow. So now you have this enclosed ribosome with spaces for the tRNA molecules to come in and read the codons of the mRNA. Okay, so it's tRNA codes or scans mRNA for the start codon AUG. And the anticodon for AUG, as we said, is UAC, right? If you have an A here, you get a U there, A there, G there, C there. You can make a song out of it. Okay, so anyway, so now we got our ribosome, which is our factory for all this to be produced. Remember that the function of ribosomes is protein synthesis, to create proteins. And here are, in this diagram, these are my little tRNAs, which in reality look more like that. They each carry an anticodon and their corresponding amino acid. So we're going to look more at this diagram here and figure out what's going on. Okay, so here's a larger picture of that same diagram. So over here, we have transcription inside the nucleus. So there's our DNA molecule. RNA polymerase is going to unzip it. Now we have our DNA template on this side. Free nucleotides will base pair to create the mRNA transcript. We put the guanine cap and the polypeptide or the poly A tail on the end. And those are going to leave into the cytoplasm. So here's our mRNA coming in with the instructions for making this protein, binding to the small unit of the ribosome, which will be scanned by the first tRNA molecule until we get to AUG, the start codon. And that is going to carry with it on this tRNA molecule the amino acid methionine. Shift, and then we get to the second 3 bases, the second codon of the mRNA. And whatever that codes for, we'd look up in the genetic code, the corresponding tRNA that can base pair with that, because of its anticodon, will come along carrying its amino acid. So if we said before, if you think back to our little example we were drawing there, after the AUG, I believe we had CAC. Let me shift up here for a sec. DNA code here. Let's say that my second codon was CCG. So I should get proline. Okay, so if my second codon was CCG, that's going to code for proline. So if this was CCG, then I would get a GGC as the anticodon on the next tRNA, and it would be carrying with it the amino acid proline. And it would bind here, and then I'd get a peptide bond between the methionine and the proline. And so I'd get this growing polypeptide chain. That guy would leave, next one comes in and shifts, and the next one comes in and reads the next codon, carrying its corresponding amino acid as dictated by the genetic code. Those two would bind, and et cetera, and they'll just keep circling in and as I get this growing polypeptide chain. And it's going to end when I get to a stop codon. Okay, so that was initiation. Let's go on down to seeing the rest of this. Okay, so here's another diagram depicting this. I get my mRNA transcript. OG is always the first one that will bind to the tRNA carrying methionine. It's going to leave. Next guy comes in, reads the second codon carrying the corresponding amino acid as dictated by the genetic code. It does a peptide bond to the first one, so now I have a 2 amino acid long polypeptide chain. This guy leaves, that guy comes in, and now this mRNA shifts to space, and I'll get the second, a third codon here, which will bring in another tRNA into the space, bond with that, leave behind its amino acid. So now I have 3 amino acids in my polypeptide chain, etc., etc., etc. This will keep happening to create this long polypeptide chain until I get to a stop codon on the mRNA. That'll stop it, enzymes will come in, cleave it, and it'll fold up into the protein that I'm supposed to get. And by the way, where do those proteins go? Well, they're going to go inside of the endoplasmic reticulum, because remember the rough ER, the ribosomes sit on it. So they'll go into the ER, eventually pinch off in a vesicle where they'll travel to the Golgi apparatus, which will then ship that protein onto its merry way, either inside the cell somewhere if it's needed, or outside the cell. So for example, if this protein was insulin, it would get shipped outside the cell to help you digest your meal. Okay, so the second part is elongation. So this is where we're going to do what we just talked about growing this polypeptide chain. So here's just another picture. There's your ribosome. Here is a tRNA stuck in there. Here is your mRNA transcript that's being read. And all these tRNAs are just going to keep coming and coming and coming as this thing moves along. So every time you get a tRNA there, its anticodon will base pair with the codon on the mRNA. And then same thing with the next one. So let's watch another video to explain this a little better. Okay, so here's yet another video on protein synthesis just to kind of reinforce. We've already talked about initiation and elongation, and we sort of talked about termination, but we'll go back over that again. So best way to explain protein synthesis is to see it in 3D.
With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to a molecule known as messenger RNA.
transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traveling in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the...
How cool is that? These 3D animations are just amazing. I wish I had this when I was in school. But you could see there, you could see how these tRNA molecules move in and how that polypeptide chain is elongating. And like it said, the last step is termination. And so this is where you get to a stop codon, and a bunch of enzymes are going to help cleave this thing. When it says molecular machine on the video, what it's talking about are certain other enzymes that are going to help to fold these guys up. And you'll get your 3D configuration of your protein there. And then that causes at the end, the ribosome breaks apart again, and it's ready to function on a different day. So let's take a final review of transcription and translation, the whole thing together. Watch one more video just showing the whole thing together again, and then I'm going to show you a classic as an addition to this lecture. I won't put it on here because it's about 13 minutes long, but it's the protein synthesis dance, which was done back in I think the early 70s, and it's pretty funny, but it actually will help. If you understand protein synthesis, you'll be able to understand that dance. But anyway, for this, just again, transcription occurs in the nucleus. DNA is unwound at the start of a promoter region, which is a short segment of DNA that signals the start of a particular gene. And that's where RNA polymerase, this enzyme, will bind. It will unzip the DNA into two sides just at that gene. It'll wind up everywhere else. But this is the gene that needs to be translated into a protein. And so one side of the DNA will serve as a template, and you'll get a base pairing using free nucleotides to create the single-stranded mRNA. And just remember that instead of thymine, RNA uses uracil. When it's done, it'll get a cap, which is a modified guanine, and it'll get a poly A tail at the end, which is a bunch of A's, a bunch of adenines stuck on there. And the introns will get spliced out. So we'll get rid of that evolutionary junk and splice together the exons, the coding regions of the gene. Then that finished mRNA transcript is going to leave the nucleus through a nuclear pore, enter the cytoplasm, and then you're going to have the mRNA molecule bind to the small subunit of the ribosome. The AUG codon, OG, is going to signal for a tRNA carrying methionine, and the anticodon to AUG is going to come and bind, and by the genetic code we know that AUG codes for methionine. Next tRNA comes into the space, it'll read the second codon, the next three bases on the mRNA, and based on the genetic code, it will carry the corresponding amino acid, which when this guy leaves will bind, that amino acid will make a peptide bond to the previous amino acid, so we get a growing polypeptide chain. That guy shifts, mRNA shifts, next one comes in, reads the third codon, etc. And so you get this growing polypeptide chain, which when you get to one of those three stop codons will stop the process, And then some molecular machines, aka enzymes, will cleave this thing off and fold it into its three-dimensional protein conformation. That protein will then enter the inside of the endoplasmic reticulum, be shipped out on a vesicle to the Golgi apparatus, which will then ship it to where it needs to go, inside the cell or outside the cell, and that's how it's done. So let's watch a final video putting this whole thing together that is included in your mastering. Here we go.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured.
Okay, so here's transcription, that first step of protein synthesis.
The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA.
This is still in the nucleus.
As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected.
Evolutionary junk.
Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases, called an anticodon, that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids.
And finally,
for proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
okay, so that is protein synthesis. I hope you understood it. Make sure you understand it because this is the basis of everything else we're going to talk about this semester. It's thanks to protein synthesis. It's thanks to the central dogma of DNA coding for a protein, coding for a trait, that evolution is able to happen. And so in our next unit, in Unit 5, you are going to actually be learning exactly how evolution happens. It starts with the molecular level, thanks to the genetic code and what happens during protein synthesis. If you have any questions, make sure you let me know and let's discuss it on the discussion board. Bye.